Awesome. Hello, everyone. Welcome back to our Tuesday seminar series on cerebellum. Unfortunately, Reza is away this week, so I will be introducing our speaker and leading our questions and discussion segment at the end. My name is Nazanin, and I'm a graduate student at Reza's lab. And today we have the pleasure of hearing from Dr. Nate Sawtell's work. Dr. Sawtell is a professor of neuroscience at the Zuckerman Mind-Brain Behavior Institute at Columbia University. He received his PhD from Brown University, where he worked with Mark Baer on experience-dependent plasticity in the mouse visual cortex. He then did a postdoctoral fellowship at Oregon Health and Science University with Curtis Bell, where he began working on cerebellum-like circuits in the electric fish. And whenever you're ready, Dr. Sawtell, please feel free to unmute yourself and begin your talk.
Great. Thanks. This is a great series. I'm happy to be able to share my work today. You guys all hear me okay? Yeah, okay, great. So I'd like to see. Sorry, it's not. Oh, there we go. Okay, so I'll start with a little bit of evolutionary background here. So as most of you probably know, all jawed vertebrates have a cerebellum in red here. And then maybe less familiar is the fact that many vertebrates also possess one or more sensory structures in their brain that share similarities in terms of development, gene expression patterns, circuitry, and function with the cerebellum. And these are the topic of my talk today, these so-called cerebellum-like sensory structures. Now, also interesting to note here is that the antecedents of vertebrates, so groups like the lamprey, for example, have these cerebellum-like sensory structures, but not a true cerebellum. So some people speculate that these cerebellum-like sensory structures may have actually been the precursors to the cerebellum. So this is one interesting reason to study them. Another is this host of similarities and differences that we're familiar with from other examples of convergent evolution in nature. So we think, and in my lab, we try to take advantage of these similarities and differences as a kind of natural experiment to understand cerebellar circuit function. So we're interested in these structures in their own right, but also, and I hope to convince you today, that we think these cerebellum-like sensory structures have something perhaps to teach us about the cerebellum and cerebellar function. So we study a couple of these structures in the lab. One is the electrosensory lobe in weakly electric fish, and that's the structure I'll focus on today. But we also study other structures with this comparative approach in mind. So in the mammalian brain, it's interesting to note that there's one of these cerebellum-like sensory structures. It's in the auditory brainstem. It's called the dorsal cochlear nucleus. So there's a host of these structures across vertebrates. And before getting into it, I want to tell you a little bit more, because this is a cerebellar audience, a little bit more about the similarities and the differences in the circuitry of these cerebellum-like sensory structures compared to the cerebellum itself, which you're more familiar with, presumably. So as you know, the cerebellum has Purkinje cells, and this critical cell of the cerebellum is characterized by the two very different excitatory input pathways that impinge on it. On the one hand, there's the mossy fiber granule cell parallel fiber system. So you know that Purkinje cells in a cat or a human or a mouse get hundreds of thousands of weak excitatory inputs from granule cells, these most numerous neurons in the vertebrate brain. So this is quite a remarkable specialization. And then on the other hand, one of the most powerful excitatory synapses in the brain, a single climbing fiber input makes many hundreds of synaptic contacts onto the Purkinje cell. So this is this singular, very powerful input. So these two very different input pathways are one of the hallmarks of the cerebellum. Another hallmark is these two distinct processing stages. So in the mammalian system, you know this as the cerebellar cortex. The inhibitory Purkinje cells project to the deep cerebellar nuclei, the vestibular nuclei, and also some connections to the parabrachial nuclei. So there's these two processing stages. And we see these processing stages in all cerebella, in all vertebrates, although the topology is not always the same. So I'm showing here the cerebellum of teleost fish, and you see here the similarities. The Purkinje cells get granule cell input and climbing fiber input. The Purkinje cells are inhibitory. They inhibit glutamatergic output cells, but interestingly, in this structure, the output cells are not located in a distinct deep cerebellar nucleus, but they're adjacent, intermixed in the same layer as the Purkinje cells. And now if we turn to the cerebellum-like sensory structures, we see some similarities that I'll draw your attention to, and also some differences. So we see Purkinje-like cells that get input from this distinctive mossy fiber granule cell parallel fiber system. And just to convince you a little bit more, let me zoom in on this. So this is a transverse section through the electrosensory lobe that we study. And what I've highlighted here is the granule cell layer, molecular layer of the electrosensory lobe, the cerebellum-like structure, and then the molecular lobe of the cerebellum proper, or a portion of the cerebellum proper of this animal. And I've shown you this here just to highlight the fact that the granule cells, the adjacent granule cells that send the parallel fibers to these structures, are one continuous granule cell layer. So the granule cells that go to the cerebellum and to the cerebellum-like ELL are closely associated with each other, even adjoining, adjacent, or continuous. And you can see some of the morphology of those cells on the left, and you can see that these are a lot like cerebellar granule cells. And we recorded from these cells, their physiology is a lot like cerebellar granule cells. So this is more than just a passing similarity. These are striking similarities in terms of this granule cell system. Cells like Golgi cells, unipolar brush cells, molecular layer interneurons, all of those are shared between the cerebellum-like structures in the cerebellum. The Purkinje-like cells are also strikingly similar, and this is perhaps most clear in the mouse dorsal cochlear nucleus. Sorry, I'm showing you a picture of that here on the right, and you can see this is staining for cerebellum. There's many gene expression similarities between Purkinje cells of the cerebellum here and so-called cartwheel cells of the dorsal cochlear nucleus, which is shown down here. And there's at least 3 independent mouse mutations in which Purkinje cells of the cerebellum selectively die, and the cartwheel cells of this dorsal cochlear nucleus selectively die. So these are striking similarities, but certainly not to say that these structures are identical. And the most striking and dramatic difference is that in all of these cerebellum-like sensory structure, there is no climbing fiber input. So the inferior olive doesn't project to these structures, but instead they all get sensory input from peripheral receptors. So in the case of the electrosensory lobe, the ELL is a somatotopic map. It gets receptors, electroreceptors that are scattered over the fish's skin. We'll talk more about that in a minute, but those are the input, the other main input to this structure instead of the climbing fiber. In the dorsal cochlear nucleus, this structure gets input from the auditory nerve. In other cerebellum-like sensory structures, there's different sources of peripheral input, but in all of these cases, these cerebellum-like sensory structures get input from some kind of a sensory map. So these are some of the similarities and some of the differences. The questions I'm going to ask today and try to answer for you are 2. So, and they're all going to be in the context of this mormyrid ELL, and we can talk a little bit about, if we have time at the end, about what this may say, if anything, for the cerebellum itself and for Purkinje cells. So what do these Purkinje-like cells transmit? What are the signals that they're conveying to this output stage? And 2, how are these signals computed? So we're going to try to get at these questions in the ELL today. And so to do that, I need to put this work in the context of the animal that we study. One of the advantages of this electrosensory system is that it's embedded in a tractable sensory motor system. And we have a good idea, because this is a sensory structure, of what the function of the electrosensory lobe is. So the ELL is processing electrosensory information. It's the first stage of processing. So its job is to faithfully transmit behaviorally relevant signals from the electrosense to the rest of the brain. But there's a challenge in doing that, and that is that this fish, this electric fish, has an electric organ in its tail. It emits its own large electric organ discharge pulses called EODs. So the fish's electroreceptors on the skin are sensitive to weak electrical fields emitted by animals in the water, like prey, for example. So this is invertebrate prey that the fish might eat, and this would emit a tiny weak electrical field. The challenge for the system is that this signal could easily be overwhelmed by the emission of this fish's electric organ discharge. The fish use these EODs to communicate and also actively sense their environment. So the EODs certainly have a purpose, but from the perspective of this passive electrosensory system, they're a source of self-generated noise. And so this is a particular example of a general problem for sensory processing that exists in many sensory systems in many animals, the so-called problem of reafference, or in other words, how do sensory systems distinguish behaviorally relevant external signals from self-generated signals. So this is an example of that problem, and we've studied for many years how it's solved. So I'm going to take you through that. So if we recorded from one of the electroreceptors on the skin, one of these passive receptors that's sensitive to prey, and we triggered that recording on the time of the EOD, we'd see this large ringing pattern of activation that lasts almost as long as the interval between EODs. So this is the source of self-generated interference that the system has to deal with. A signal related to the prey might be buried in this large firing rate modulation. And what makes this problem even more difficult is that this EOD-induced interference isn't fixed or constant throughout the life of the animal, but will change on different time scales. So in the African rivers where these fish live, during the rainy season, the water levels rise, the conductivity of the water changes, and that affects the amplitude of the EOD. So that's what's shown here. So at some times during the season, the EOD will cause a very large pattern of activation, at other times very little. On faster time scales, this EOD pattern could change depending on where the animal is in the water. So if it's near a non-conductive boundary or if it's an open water, this EOD-induced pattern could also change. So this is not a problem probably that could be solved by an absolute fixed gating. You might need a more sophisticated mechanism, a mechanism for predicting and canceling this self-generated interference. And this kind of mechanism is exactly what's been shown and studied for some years at the first stage of processing in the electrosensory lobe. And at the core of this mechanism is the integration of sensory input from the periphery, from electroreceptors, with an additional stream of information. And this is a copy of the motor command to discharge the electric organ. And these motor copies, sometimes they're called efference copies. I'll call them corollary discharge today. It's the integration of these two signals in the electrosensory lobe that is critical for solving this problem of reafference for the electric fish, and is critical for what I'm going to tell you about today. And we can understand, and we do understand much more specifically what this corollary discharge signal does. And I can schematize it for you on the right, and then we're going to talk about this, the mechanisms of this, for the rest of the talk. So it's important for that to be clear. If it's unclear, please feel free to ask me. So again, imagine you're an electric fish swimming around in the water, and now we're going to eavesdrop on the response of one sensory receptor, and we're going to look at the activity of that receptor. And if we diagram in the times of the fish's electric organ discharge pulses, we'd see these large patterns of activation. I showed you those earlier, and then buried in there somewhere might be a signal of interest, but we can't quite see it. So in principle, what you'd want to do is use the corollary discharge signals, this additional stream of information that's correlated with the fish's electric organ discharge pulse, and use that to make an inverted copy of the effect of the EOD. So that's the flipped EODs here, an opposite pattern, and simply subtract those two signals. And if you were able to do that really well, what you would reveal is the behaviorally relevant signal of interest, this prey diagrammed in here in green. And you'd send this important behaviorally relevant signal to subsequent processing stages. So this is what we would like to happen in the ELL. And I'm telling you this, of course, because we have lots of evidence that this, in fact, is what happens. And I'll show you some of that evidence and talk about the mechanisms. But it's worth, before moving on, that this is relevant to the cerebellum, because this idea that the ELL generates and subtracts predictions of the sensory consequences of motor commands is an example of what many people think the cerebellum does. So this is a diagram from Daniel Wolpert and Chris Miall, and you can find many similar ideas out in the cerebellar literature, that the cerebellum is somehow an internal model or a forward internal model that predicts the sensory consequences of motor commands. And there's many possible uses for such predictions, but one of them is to cancel reafference. And that's exactly what we see in the electrosensory lobe. So let's get into it now. What are the cellular and circuit mechanisms whereby this can happen in the electrosensory lobe? And we've studied this for many years, and we've used computational modeling, recordings in vivo and recordings in vitro, and tried to put all these things together. And I'll tell you about that, but I'm also going to tell you about kind of a new tool in our arsenal, and I'll frame the work in that way, and that is a connectome of the ELL. So this is a project we've been working on for several years now, and if you're interested in this workflow, it's quite an amazing process and developed by many talented people that we've had the opportunity to work with. So I won't go into the details of how we got the data, but I'll show you the results. But it's quite an amazing technology, and I think the fact that we've been able to use it in this small system is kind of a testament to how increasingly accessible this kind of work is, and how it could be used, I think, in many systems to address questions in neuroscience. So we're very appreciative of all the help we've gotten from Lichtman Lab and Google and others in putting together this data set. And this has really been driven by a really talented postdoc in the Engert and Lichtman Labs, Mariela Petkova, and a research scientist in my lab that's really done an amazing job analyzing this huge data set. So what do we get out of this connectome, and why is it useful in the ELL? And the short answer is that in the ELL, we have a lot of computation packed into a small space. So the limitations of this connectome work is it's incredible high resolution, but it's costly and it's hard to do this for large volumes of tissue. So the ELL is really perfect because you have a lot going on in a small space. And so I'll show you that here. So if we zoom in on a slice of the ELL, and our volume, it's about 400 by 400 by 150 dimension in microns. So that's about what you're seeing here. And what you can see is we have the sensory input from the periphery in green. So this is the electroreceptor afferent axon coming in. So this, we understand what this is. This is the sensory input from receptors on the skin. And on the other hand, we have the corollary discharge input. So this is the mossy fiber granule cell parallel fiber system that makes this a cerebellum-like circuit. You can barely see these tiny thin axons. They're thin unmyelinated axons like in the cerebellum, and they synapse on the spiny dendrites of the principal cells of this cerebellum-like sensory structure. And then, as you alluded to earlier, we have these two processing stages. We have Purkinje-like cells that are GABAergic, and these synapse locally here onto glutamatergic output cells. So the analogy here is to Purkinje cells and deep cerebellar nuclear cells, but conveniently here for us and for this connectomic approach, these cells are intermixed and adjacent to one another. So their synapses are all local here and all contained within the volume. So using the connectomics, we can work out the connectivity of the two major input streams and these major players, the Purkinje-like cells and the output cells, all at the same time. And I should say that another critical aspect here is we're able to make sense of this data is because in past work we've recorded intracellularly the physiology of these various cell types, and we can identify the cell types based on their morphology. So we have lots of data from past work that we can then superimpose on this connectome, and we're able to make that connection because the different cell classes we've recorded from are morphologically identifiable. So I won't take you through all the details of this, but in this connectome, we can look in great detail, of course, at the morphology of the various cells and put them in categories that map onto the categories in the past work. So this is an example of two of these Purkinje-like cells that fall into two categories, and you can see they split very nicely based on their morphology. So we have a strong ability here to link up the cell types that we see in the connectome to a vast body of about 50 years of prior work, many, many recordings from this brain structure in vivo and vitro from identified cell types. So these are the tricks that we're going to use. And I'll just tell you a little bit more about this past work and the physiology, because it's critical to really understand the phenomenon that we're trying to describe. And this is another question is, why do we study the system in the first place? And the short answer is, in this system, we have really compelling neural correlates for predictions of the sensory consequences of the animal's own behavior. So let me show you what that means here. So in this system, when we immobilize our preparation and we record, the advantage that we have is that the sensory input to the system is blocked. Okay, so there's a motor sensory loop. The fish is emitting this electric organ discharge, this simple motor act, but when it's mobilized, the emission is blocked, but the fish continues to emit the motor command to discharge the electric organ. So the corollary discharge system, this input that comes in through the granule cells, this input system is intact, and we can study its effects in isolation. So in many cases, in many systems, we know corollary discharge exists, but it's like, imagine reaching, for example, in a human, the corollary discharge is there, but how do you isolate its effects in the brain? How do you separate it from proprioceptive or visual feedback? So that problem is circumvented in the system because the sensory consequences are very simple. They're electrical pulse, they're completely blocked. So the corollary discharge can be completely studied in isolation in this system. And then the second trick is that we can not only open this loop, but then close it back up again. And so we can now record this corollary discharge response, monitor it, and then at our will, we can replace the sensory consequence with one that we control. So we can deliver an electrical pulse, we can change its timing, we can alter its polarity. So we can control very easily the sensory consequences of the motor command in the system. So we can try to teach the system new sensory consequences. We can look at the sensory effects on their own, and we can probe the effects of the corollary discharge before and after manipulating sensory consequences. So let me show you some of the results of these experiments, and that kind of sets up what we're trying to explain. So here's a typical experiment. This is a recording from one of the output cells of the circuit. These large fusiform cell is the name, and I'll try to use color codes and remind you of the names, but the names are going to become important here. This is one class of the output cell of this structure. And so if we record in this cell, we can start by looking at the response to the corollary discharge alone. So this is a histogram triggered on the time of the corollary discharge, and we see not much response. In the second phase of the experiment, we pair that corollary discharge with an EOD-like stimulus, and we see that strongly excites the cell. We call this an E cell because it's excited by the EOD, E for excited or excitatory. But as the experiment goes on, and now we wait 10, 15 minutes, we see that the response, this initial strong response, is strongly diminished. So something has changed here. And if we turn the stimulus off at this point and return to this original condition, we see that the effect of the corollary discharge has changed, and it's changed in a striking way, and that now it's a temporally specific negative image or inverted copy of the effects of the sensory stimulus. So just in 10 or 15 minutes, the system has learned a prediction of the sensory consequences of the Fish's own motor command. And we call this the negative image. We can do one more trick and just play the sensory stimulus again alone, and we see that it still evokes a very strong response even after all of this pairing. So this is not a case of fatigue or adaptation of the receptors. This diminishing response here that we call cancellation is a result of this negative image. So this is our neural correlate for a prediction of the sensory consequences of motor command. Here's one version, and now we can do the same exact experiment in the companion output cell. So there's two classes of output cell. This one's called the large ganglion cell, and you see the same pattern with the difference here that this cell class is inhibited by the EOD. So this is kind of like on and off channels in the retina. There's output cells that are excited by the sensory input, output cells that are inhibited, but in both cases, we see this cancellation of the sensory response as it becomes predictable, and this generation of this temporally specific negative image. So this is what we want to understand. How are these negative images generated? How are they formed in this circuit? And we think that if we could understand this, this would be interesting because, again, this is an example of the idea, the general idea that cerebellar circuits could generate predictions of sensory events or predictions of the sensory consequences of motor commands. Okay, so what will we need to do this? These negative images are temporal patterns that are clearly modifiable by experience. So we would think that we need at least two things. We need some kind of temporal representation, a temporal basis set, we could call it. And then we need some way to modify the response. And so we can look for these two things. And this is work we've done in the past. We recorded from the granule cells and asked, is there a response? Is there a response that spans time? So these negative images span around 200 milliseconds. The input related to the corollary discharge, so the motor command itself, is only about a millisecond. So it's not a foregone conclusion that the granule cells would respond spanning this 200 millisecond delay, but this is what we found when we recorded intracellularly from many of these granule cells. And this is work from some time ago, but when we record it intracellularly, we see different temporal patterns of activity in the granule cells that could potentially form a temporal basis set for sculpting negative images. And then also known from past work is that these synapses are plastic. And this is work of Curtis Bell from the 90s. And what he showed in one of the first demonstrations of spike timing-dependent plasticity, in fact, in the brain, is that the synapses between granule cells and these medium ganglion cells exhibit a kind of plasticity where if a presynaptic input comes before a postsynaptic spike, that synapse is weakened. Inputs coming in at different delays are strengthened. So this is the opposite of Hebbian plasticity, where the cells that fire together wire together. It's this anti-Hebbian plasticity, and you kind of recognize this as in some ways similar to long-term depression in cerebellar Purkinje cells, so kind of LTD. So these are key ingredients, and now we can zoom in a little bit more, look at where this is actually happening, and what we can see here, and what I want you to what to emphasize here is that the vast majority of this plasticity is happening at synapses onto the medium ganglion cells, onto their spiny dendrites. So you think about the Purkinje cell as getting hundreds of thousands of parallel fibers and being the main reservoir of granule cell input and plasticity. The same thing seems to be true in the electrosensory lobe. However, it's also true that there are some spiny apical dendrites of output cells, and you can think of this as analogous to the fact that in the cerebellum, the deep cerebellar nucleus also gets some mossy fiber input. So two copies of this information are sent to these different processing stages, but seemingly in different amounts. So I'm going to focus on this plasticity onto the Purkinje-like cells and on two hypotheses. One, that negative images are formed by anti-Hebbian plasticity at granule cell synapses onto MG cells, and then subsequently transmitted to the output cells. And so we'll focus on the second, the transmission, first, because that's a bit easier. So to ask about this, just remind you about the two different sensory responses that we see in these two types of output cells. So if we record from the large fusiform cell, we see that it's excited by the sensory input. This is the membrane potential of the cell. It's depolarized. If we record in the large ganglion cell, we see it's hyperpolarized, so on and off responses. So this is just to say that we need two different kinds of negative images to cancel these predictable responses. And if we record from the medium ganglion cells, we see indeed that we see two different responses. And now these are responses to the corollary discharge after we've performed these pairing experiments. So after the system has learned, we see that the corollary discharge responses have come in these two flavors. So this leads us to a clear hypothesis that we can test with the connectome, and that is that these MG1 cells, one class of MG cell in blue here, should inhibit the large fusiform cell. So this is an inhibitory connection. So if the MG connected to the LF cell, it would oppose this excitatory response. And then conversely, the second class of MG cell, the MG2 cell, should inhibit the large ganglion cell. So is this actually correct? So to test this, we can use the connectome. I'm showing you two example cell output cells, one of the large fusiform type, one of the large ganglion type, and we can look now at all of the MG cells that connect and inhibit these cells. And if we do that, we see that these clearly separate into these two groups, matching the connectivity that we hypothesized. So all of the MG cells in this particular, that synapse onto this particular large fusiform cell are of the MG1 type. All of the MG cells that synapse onto this large ganglion cell, example on the right, are of the MG2 type. We also found characterized connectivity between the MG cells. And we look here and we see that all of the synapses onto the MG2 are from MG1. All of the synapses from MG2 onto MG1 are from MG2. So again, striking patterns of structured connectivity, highly non-random. This is the matrix for all of the MG1 and MG2 cells that we reconstructed in our volume. And then we can ask, since we know the physiology, we can ask using modeling what this means for sensory cancellation. You can already see from what I showed before that the connectivity between the MG cells and the output makes sense, is critical. What about the recurrent connectivity? We can test that in the model. And what we see is that the rate of cancellation, which I'm plotting here, is much faster if we include the network connectivity that we observe. It's sped up by the MG cells, and then it's sped up even further if we make those MG cells recurrently connected to one another. So the connectivity that we've described so far makes sense. It allows the MG cells to transmit negative images that are appropriate for canceling sensory input and output cells. But kind of an even more difficult question, comes up if we really think more about how these negative images are actually formed to begin with. And so to highlight that, I need to take you back a little bit and tell you a little bit more about some of the theory of the negative image and of how the negative image could arise from anti-Hebbian plasticity. And this is work that was done very nicely in the 90s and 2000s by Patrick Roberts and Curtis Bell. So let me take you through that real quickly. So how do we think about this anti-Hebbian plasticity? So as I told you before, the anti-Hebbian rule tells us that if a presynaptic input comes in before a postsynaptic spike or is associated with an increase in postsynaptic firing rate, we get weakening. So you can see that schematized here. This is the effect of the EOD on the firing rate of an output cell. For periods when that firing rate is elevated, the plasticity rule tells us we'll get an increment of weakening in the granule cell input. That will subtract from the response. Now we see the cell is firing less than its equilibrium rate. We'll get an increment of strengthening. And as that process happens over and over again, we end up with an equilibrium firing rate that's flat, and we get this negative image. So this is just, again, to show you that this anti-Hebbian plasticity is an appropriate learning rule to generate negative images. The perplexing thing, though, is that while this negative image can form based on the anti-Hebbian plasticity in a cell-autonomous way, it doesn't seem to be a mechanism for transmitting a signal to a downstream cell. So if this is the process we believe is happening inside an MG cell, we're left with a puzzle. How is the MG cell transmitting the negative image to the output stage of the network? So I've shown you that that's what is happening based on our recordings, and I've shown you connectivity that support that. But if we really think about it, it really doesn't make sense given what we know about the plasticity rule. So this is the next piece of the puzzle that we wanted to try to resolve. And I think the answer to this will be familiar to you, but with some twists, and that is that these medium ganglion cells, these Purkinje-like cells, fire two distinct types of action potentials called narrow and broad spikes. And when we record, making a long story short, we see that the broad spikes encode sensory input, and we also know that these are the spikes that induce synaptic depression at granule cell synapses, while the narrow spikes transmit the negative image. And you'll kind of be reminded here of the situation with Purkinje cells, where you know that Purkinje cells also fire two types of spikes. They have complex spikes driven by input from the inferior olive and simple spikes. Simple spikes transmit the main output of the Purkinje cells. Climbing fibers induce synaptic plasticity that shapes that output in some way. So there's some interesting, clear analogies here. The interesting, puzzle here, and the difference is that whereas in the case of the Purkinje cell, we know very well where these complex spikes come from and how they're separate from the simple spikes. So the climbing fiber is generated by this powerful excitatory input. There's a separate brain structure, the inferior olive, that generates these complex spikes. But what about the ELL? I've already told you that in the ELL, there is no climbing fiber input, and yet we still see this separation of responses and separation of function for these two types of spikes, the narrow spikes and the broad spikes. So how does this separation arise? That's the question then. So I'll show you kind of a phenomenological model supported by the data, and then I'll tell you how we think this works, and then we'll go back one more time to the connectome. Okay. So this is one half of our circuit, an MG1 cell to a Purkinje-like cell to a large fusiform output cell, and I've already told you that the large fusiform cell is excited by sensory input. And then after learning, its response is canceled. So the solid lines here are after learning, the dashed lines are before. So this is what the output cell does. This is the desired output, and what we need is for the MG cell to be able to transmit a negative image. So it's going to transmit a response with similar shape as the sensory response of the large fusiform cell. It's going to do this based on anti-Hebbian plasticity of the granule cell synapses. And here's what we actually see in the responses of this cell. So if we record from one of these MG1 cells, we see that broad spikes, so the spike that's inducing the plasticity, is inhibited by the sensory input. We see initially that the narrow spikes don't respond very much. After learning, we see the broad spikes are canceled. So this cancellation process is familiar, and this is the expected result of the anti-Hebbian plasticity. We see a negative image. The corollary discharge response to the MG cells formed a negative image of the response to the sensory input on the broad spikes. And then, importantly, as we desire here, now we see the narrow spikes are conveying a negative image, or a response that would be appropriate to cancel the sensory input to the large fusiform cell. So this is what we want. Broad spike. The broad spike output is the sum of the sensory input and the corollary discharge. Narrow spikes are transmitting this corollary discharge signal, and that gives us the cancellation at the level of the output cell. But still, there's a big puzzle here. If these two inputs are canceling each other out, how are we transmitting the negative image? So we haven't yet solved the problem. We just kind of cartooned it again. And so this is work of Solomon, Mueller, and Larry Abbott. And what he was able to do is to build a biophysical model and really trace down the mechanism of this separation inside the medium ganglion cell at the level of the generation of the broad spike. And so what Solomon came up with is the following. So how does this actually work?
Nate, question?
Yeah.
Hi, Nate, I'm Farzan. It's good to
Hi, how's it
Yeah,
going?
it's good to see you. Good. Yeah, so great talk, very clear. A few things, because I keep thinking about mouse and it's a bit hard to, I forget which one was which one. So this is a great slide to make sure. So MG1 is like the Purkinje-like is MG1?
That's right. Yes, that's
Okay, and LF is like the
one of
cerebellum.
the output, exactly. Half, yeah, there's two types of each, so it's a little confusing, but this is
Right.
one half of our circuit.
And then it seems like the error computer, like the inferior olive here, is combined with the output, if I understood things correctly, because it seems like the one that receives both the prediction and the actual sensory input and computes the difference is the output. Kind of.
Yeah, they're kind of both. Yeah, this is what's kind of interesting here. They're
see.
both.
So it's not like we have a separate inferior olive and a separate that is computing the difference, and the scene is sending the.
Yeah, that's right. The difference this difference is being computed inside of these two cells. Both of these cells are integrating these two streams of input. I'm emphasizing what's happening in this medium ganglion cell here and showing you how it can do that computation. But it's also true that that computation can be happening inside of this cell itself.
Right, which is also for mice like the DCNR, they do talk about it, that they receive the
climbing fiber collateral
exactly.
and mossy fiber.
Exactly, yeah.
You can map this, yeah, exactly. So anatomically, there's a parallel in that both cerebellar cortex and nucleus receive these two streams of information. And if you want to map this on, you would map on the sensory input to the climbing fiber, although, of course, the sensory input is not the same as input from the inferior olive in many ways. And
then because there we think climbing fibers are teaching signals, but here there is no such, there is no, so what drives learning is still.
Yeah, so so the learning here is driven by the sensory input that induces the dendritic spike. So whether we might not want to call it we can call it a teaching signal. It may be a different kind of teaching signal, but it's driving plasticity. It's driving heterosynaptic plasticity at another site. So it's similar to the cerebellum in that way.
Right, but I guess the question is that the sensory input will not change over the course of learning.
Yeah, yeah. Yeah, exactly. So that's interesting. So
Right.
cerebellum, you know, if we think of the cerebellum as error-based learning, the climbing fiber induced error may be changing and diminishing over learning. Here,
Exactly.
this is a fixed input. So some people might call this target-based learning. So it's a
Mm,
kind of sort of like supervised flavor learning, but instead of an error-based system, you could call this a target-based system. That's a
I
term
see.
that
But
we've
then,
used. It's a term that Jeff McGee actually uses now for CA1 hippocampus. So maybe
mm,
we can talk about this a little more at the end. I
sure,
have
go
a
ahead.
few
Yeah,
more slides that we
thanks.
can
Yeah,
talk about. I think it'd be fun if there's time to talk about
sure.
similarities and differences and how we
Yeah,
conceptualize some of these things.
thank you.
Okay, so the problem that I'm trying to motivate here is, in the cerebellum, we have an inferior olive that computes this error signal and can explain how the broad spikes, how the complex spikes and the simple spikes are doing separate things. How do we explain this separation in the ELL? And it's a bit more subtle, and we can trace it down to the biophysics of spike generation in these cells. So what happens when the sensory input comes in? What Solomon showed in the biophysical model of the MG cell is that sensory inhibition can reduce the narrow spike, can reduce the broad spike rate by reducing the amplitude of the narrow spikes. And so what we discovered from this biophysical modeling is that the proximal stimulus for triggering a dendritic spike is actually the backpropagating axonal spike. So there's a biophysical coupling between the axonal spike and the dendritic spike. And by controlling the amplitude of the axonal spike via inhibition, we can control the rate of the broad spike without affecting the rate of narrow spikes. So that's the critical separation we need. We need to control the rate of these two spikes separately. And we do that by controlling actually the amplitude, or you could think about it as the efficacy of the backpropagation of this axonal spike. So that's what's schematized here. By decreasing the axonal spike, we get further from broad spike threshold. We decrease broad spike rate. So that's this dip in broad spike rate, but we do that without affecting the narrow spikes. So this decrease in the broad spike rate triggers potentiation of the granule cell inputs. So that potentiation depolarizes the granule cell membrane potential and gives us an increase in the narrow spike rate. And that increase in the narrow spike rate is the negative image that we need to transmit to the output cell. So this little nifty mechanism here can explain how we can essentially have this anti-Hebbian plasticity, have a cancellation and a stable learning at the level of the broad spikes, but at the same time, have a signal left over to transmit. So we think this is quite an interesting and clever mechanism that this medium ganglion Purkinje-like cell has arrived at. And I'm not going to show you the cartoon, but we can explain this also at the MG2 cells on the other side of the circuit, because again, we need two separate negative images here. We can explain that with disinhibition. So here we have synaptic inhibition. If we replace this inhibition with disinhibition, then we can get an increase in the broad spike rate and a decrease in narrow spike output. Critically, though, the sensory input has to be relayed via inhibition and disinhibition, and that's because we need inhibition to selectively control this axonal spike amplitude. If we have excitation, we could alter the narrow spike amplitude, but we would also be affecting the narrow spike rate because we're so far from the equilibrium potential. So we couldn't get this selective effect on the amplitude without affecting the rate with excitation, only with inhibition. So this is what we learned from the biophysical modeling. This is what we have some evidence actually in vivo for these changes in the axonal spike amplitude. But the big question then, and what's going to take us back to the connectome, is whether indeed the sensory input is actually relayed via inhibition and disinhibition. And so far what I've showed you is these are excitatory electrosensory afferents. You can see them synapsing or potentially synapsing on the basal dendrites of these cells. Is there actually any evidence anatomically for these inhibitory and disinhibitory pathways that the model seems to absolutely require? And so we can address that by tracing backwards from the MG cells. Now we're going to go backwards. All of the synapses, excluding the apical dendrites, all of the somatic and basal dendrite synapsing onto the MG2 cells in blue, the MG cells and the MG1 cells in blue, MG2 cells in red. And what we see is really a striking picture is indeed there are many additional cells. So we don't just see electroreceptor afferents, we see a few afferents when we trace back, but the majority are all of these other cells that we really didn't even know for sure existed, but the model clearly predicted. And of course, those need to be inhibitory, and we are working on that. We're not 100% sure, but we're pretty sure that the majority of those cells are inhibitory. So there's copious substrate here for a disinhibitory pathway. This is the schematic that we have so far based on the connectivity that we've traced. This is based on a subset of the cells in the volume, so there's still some more work to do. We want physiological evidence for this. We want to confirm the neurotransmitter types more carefully, but so far it's pretty clear from the connectome that the anatomical substrate for these prominent inhibitory and disinhibitory pathways indeed exist. So I think to conclude here, let me just highlight and then open it up to discussion here. Similarities and differences. So what have we learned over the years from these studies of the ELL? So I think we have a clear example in the ELL, neural correlates, learning rules, anatomy, all of that that shows us the mechanism for generating learned predictions of the sensory consequences of motor commands, which is a hypothesized function of the cerebellum. And I should also say evidence for this has been provided for other cerebellum-like structures. So I showed on the first slide a bunch of cerebellum-like structures. This formation of negative images has been described in sharks, other species of fish, some evidence also in the mammalian dorsal cochlear nucleus. So it could be that across phylogeny, across these cerebellum-like sensory structures, this idea of generating learned predictions is a common recurring theme. We have a function for granule cells in this circuit, which aligns well, I think, with kind of historical ideas about granule cells in the cerebellum. And those are kind of, I think, simple and still compelling ideas that these large number of granule cells could provide a basis set for learning. In particular, in our studies of the ELL, we see a temporal basis set. So we see a diverse temporal patterns conveyed by these granule cells. In the Purkinje-like cells, our work highlights key roles for the two types of spikes in Purkinje-like cells. So the origins and the details of these spikes are not identical to the cerebellum. So in the ELL, the broad spikes are sodium spikes. They're not generated by an input from the inferior olive, and yet their separate control is absolutely essential to the function of this circuit and to the generation of predictions. So this is kind of an interesting example where the function is similar, but the way it arises is actually quite different. And then finally, I think a similarity are these two different processing stages. So we talked about this at some length, but just to say it again, in the cerebellum, we have a cortex and a nucleus. Both get input from mossy fibers, either indirectly through granule cells or directly, and both get input from the climbing fiber, either as the powerful climbing fiber itself or a collateral. And then we see that echoed in the ELL, where we have these two processing stages, Purkinje-like and output cells, both of which receive granule cell input, both of which receive sensory input. So one of the questions that we're very interested in, and which we absolutely don't know the answer to yet, and would be interested in thinking about also, what do we know about this in the cerebellum? Getting your thoughts is why do we have these two processing stages that get seemingly similar information? How do we think about that? And then also some differences that are striking and equally kind of worth attention. One of them that is, kind of, interesting and obvious is just this, what you've seen and may strike you is just how many additional cell types there are in the electrosensory lobe. So we were very surprised when we traced back to see all of these many, these disinhibitory and inhibitory pathways. So clearly in this sensory structure, there's lots of additional cell types that are not present in the cerebellum itself. And and we see this across the cerebellum, like sensory structures, that there's core conserved features, molecular layer interneurons, granule cells, Purkinje-like cells, but lots of extra features that may be specializations for sensory processing. And then perhaps, of more interest to this group and more fundamental, how do we think about similarities and differences between the sensory inputs to these cerebellum-like sensory structures and the climbing fiber input, and this kind of leads to, I think, still ongoing and very important questions about what does the climbing fiber do? How do we think about it? Do we think the climbing fiber is an error signal that conveys some deviation from a desired state and then goes away? As performance is improved, do we think about it in some other way? So there's lots of data, as you guys know, that climbing fibers don't always apparently signal errors. They may always be there, even when you're performing behavior well. Sometimes the climbing fiber looks more like a motor command. Sometimes the climbing fiber is encoding reward-related signals. So there's a, kind of a, multiplicity of ideas, as well as long-standing debates about the climbing fiber. And I think that's still an open question. So it's interesting to think about whether anything that we learn from cerebellum-like structures, including this idea of a target versus an error signal, could be relevant to the ongoing question of what the climbing fiber signals and what its consequences are for cerebellar function. All right, so happy to take questions. Want to thank all the people involved, especially all our collaborators that helped and made possible this connectomic efforts. Always, great collaborations with Larry Abbott and Sullivan Mueller, really driven the ideas forward. And then just one last slide for the questions I'll leave you with. One of the fantastic aspects of this fish, and the reason why, one of the reasons why it first came into neuroscience is actually not the cerebellum-like structure that I've told you about all of today, but actually the cerebellum itself. So this fish has the largest cerebellum of any vertebrate on Earth, one of the biggest brain to body mass ratios of any animal. So the early anatomists called this the gigantocerebellum. So you see on the left, the brain of a trout, on the right, the brain of one of these elephant-nosed fish that we study, and everything you're seeing, the entire dorsal surface is covered by this immensely hypertrophied cerebellum. In the sagittal section, everything above the dotted line is cerebellum, except for this ELL back here. All of this is proper cerebellum. So this is a little eye candy, fantastic cerebellum of this fish. Someday we'll try to understand that as well. So if any of you are interested, let me know. Okay, thanks.
Thank you so much for the amazing talk, Dr. Sawtell. So if anyone has any questions, please feel free to raise your hand and unmute yourself. Abigail, please go ahead.
Hi, Nate. Fantastic talk. That was so fun. I feel like I have 100 questions and I don't know how to prioritize them, but I guess that maybe if I may sort of dive in to a broad question, do you have a sense of the or could you teach us a little bit about where the output neurons, the fusiform cells, go and what they're thought to be doing? Are they driving prey capture, or what is the sort of, besides sensory interpretation, what is the sort of computational goal of the structure?
Yeah, that's a good question. Yeah, so the ELL is the first stage of sensory processing. So all of the electroreceptor input has to go through these LF and LG cells to get to the rest of the brain. So, think about it like the retina or like the cochlear nucleus or the olfactory bulb. It's the obligatory first stage. If we knock out, if we mess up the negative image, prey detection is messed up. But there's many additional stages. So these cells are far from the they're not driving, they're not directly going to motor structures, they're going to the next stage of processing and the next stage. So they're the first processing stage in a cascade of processing stages. And so the way I think about it is, these cells are trying to report first pass. These cells are trying to report to the rest of the brain, something unexpected was in my receptive field. And, probably it's a bit more complicated than that. They may do some more feature extraction or something else, but first pass is that this is a humble sensory processing stage. It's just trying to tell the rest of the brain that something unexpected was there. And so I think of that, this is very much like, predictive coding in some sensory system where you're trying to forward to the rest of the brain whatever is newsworthy and unexpected by canceling out, predicting and taking away whatever you can.
Great, that's helpful. Now I'm going to go like real, just mechanistic. So in your beautiful reconstructions, I didn't see the molecular layer interneurons in the what I imagine is the molecular layer,
Yep.
but you've alluded to them before, so I assume they exist. And the reason I'm asking about the molecular layer interneurons is because in your discussion of the generation of the negative images, I feel like in the cerebellum proper, there's a sense that the feed-forward inhibition mediated by inhibitory molecular layer interneurons is integral to inhibition of a spontaneously active cell type, the Purkinje cells. So I'm wondering about the mapping of, first of all, whether the MG cells are spontaneously active, whether there are feed-forward inhibitory circuits through the parallel fiber-like inputs to them, and how those circuit elements sort of play into the biophysical models that you have on the generation of the negative going responses in those cells. And then I'll shut up. So anyway, beautiful, so fun.
No, this is the fun. Yeah, this is the fun part is the questions. I wish, yeah, could just. Yeah, so those are good questions. In general, you know, we've neglected the MLIs, but now that we have the connectome, you know, we get to unneglect them because we see them all and we can completely map all their inputs. There are tons of them. They're prominent. They're there. So, you know, one of the big questions, so, you know, there's good evidence, as you know, in the cerebellum that the MLIs themselves, you know, have some interesting plasticity. So some of the MLIs at least seem to get some input from the climbing fiber. And like the old studies of Jorntal and Eckert seem to suggest, right, that they're doing their own, they're like a little Purkinje cell doing their thing, their own granule cell plasticity. So whether that happens in the ELL, we don't know. The MLIs don't have any, our MLIs don't have spines. There is some evidence that they could get some sensory input. So there could be an analog of this climbing fiber collateral spillover, but we don't know. So we don't know is the short answer. If we record from one of these MG cells and we activate it with, if we evoke a dendritic spike in vivo, we see a depression of the response below, you know, so the cell is, you know, fires less and is depressed. And so we typically interpret that as LTD of parallel fiber synapses, but you could say, how do you really know that's LTD of the parallel fiber synapse? How do you know that that's not plasticity of MLI onto the MG? We don't know, but it probably isn't, though, plasticity of parallel fiber onto MLI because we only evoke the dendritic spike in the MG cell, if that makes sense. So that's kind of, you know, so the short answer is we don't, you know, we're going to look more into MLIs, but that pairing experiment, yeah, I don't know. I can't say whether we can't say from what we're doing whether it's really removal of excitation only or also potentiation of inhibition. We can't say. We tried to do conductance measurements at some point, but not clear. So MLIs are, you know, very interesting on the table to be investigated.
Arizona, go ahead, please.
Sorry, Nate, I think I missed this part of your talk. So the sensory input, we talked about that target base versus error base, right? So we do need that. So if the sensory input is constant, then the learning keep happening. How does this stop?
Right. Yeah. So that's, you know, in this system, that's beautifully solved by this anti-Hebbian plasticity, right? That is that the equilibrium for this anti-Hebbian plasticity is negative image and cancellation. So that's one of the beautiful things about this learning rule is that the sensory input is always coming in, and this learning rule keeps changing synaptic weights until it has achieved cancellation based on the negative image. And like the simple original scheme was that was a cell autonomous process that happened just inside of the output cell or the MG cell. And then I tried to explain, and I probably did a poor job, of how that can't really be the case. It can't be cell autonomous because the MG cells are actually playing a key role in transmitting the negative image to the output cell. But then with Solomon's work and showing this biophysical mechanism that separates the MG axonal spikes and dendritic spikes, we've again solved that problem and shown how we can both have stability from the anti-Hebbian plasticity and still transmit. That's the kind of the cool thing, is because you're absolutely right. You need that stability, but we have our cake and eat it too with this mechanism that Solomon described in the MG cell, where we can have the broad spikes cancel and shape the negative image, but still transmit that signal to the output cell. So that's our current understanding is, is that that's how it works. And in our models, it works in this way. And then if you take that input away, if you ever take the sensory input away in our model, in what we think we see, is that it just disappears. The system learns a sense. If you take it away, then you're teaching the system that there is no sensory consequence, and then everything goes back to a flat line. And then if you put it back, it goes back. So, in this kind of target-based scheme, the target has to always be there, or else everything is forgotten. And Solomon would argue, and that is also true in the cerebellum, and that's kind of interesting. So what happens in the cerebellum if you take away the climbing fiber input? And so you can ask that in the VOR. What happens in the VOR if you take away, if you do the VOR training, then you put the animal in the dark, then at least in some cases you forget it, right? It either gets transferred to the nucleus or you forget it. Right? The learning is not stable in the absence of the climbing fiber input. So, you could argue, you could look at the literature and make an argument that also in the cerebellum, you need the climbing fiber to maintain learned patterns in the cerebellar cortex. So that's kind of, some people would, argue with that, but it's an interesting thing to think about, whether if you look at the cerebellar literature, do you also get this idea that you need the climbing fiber to stick around? There's lots of the smooth pursuit is an example of that. If you look at smooth pursuit eye movements, even when the animal's doing it as well as it can possibly do, you see maintained antiphasic patterns of simple and complex spikes. And this is one of the oldest findings in the cerebellum. You record in the cerebellum and you see these antiphase patterns. Those aren't only there when there's errors, they're just there all the time. So you could take that and interpret that in light of this, in light of the ELL, kind of as hinting at, they're there maybe for a reason, and that reason could be to maintain appropriate patterns of simple spike output through continual plasticity. That would be our ELL, ELL-inspired view of what's going on in the cerebellum, that the climbing fiber is needed to continuously maintain these patterns, analogous to the idea that the sensory input in the ELL continuously maintains the negative image.
Thank you.
Hey, great talk. Thank you. May I ask a question?
Of course.
So this idea of the climbing fibers forming the activity. So I'm wondering, like, as you just mentioned, if we remove the climbing fiber and the response collapses to like a steady line, or vice versa, how do you justify the downstream projections of Purkinje cell-like or Purkinje cell? So if a Purkinje cell is not connected to the relevant region, even if I'm able to produce a complex spike that change the pattern relevant to a behavior, how can it transfer it to do anything in the brain? Does it make sense?
Okay. So, sorry, say it one more time.
So what I mean is that if the climbing fiber is forming the response of the Purkinje cell-like or Purkinje cell activity, what about the downstream of the Purkinje cell? Because that's firing, those synaptic connectivities are not affected necessarily by the climbing fiber. So how does the circuit make sure that the climbing fiber that is projecting, that is forming this Purkinje cell, is for the relevant movement or behavior or whatever?
Okay, let me restate it just to make sure. I think what you're asking is like the very fundamental question of, in this multi-stage, multi-layer, multi-stage learning process, how do you make sure that the plasticity at the Purkinje-like cell is even doing remotely the right thing to affect the output? Is that what
exactly?
Yeah.
And if you change the climbing fiber for a Purkinje cell, and even if the pattern changes, how do you make sure that the downstream connections are also changed in a way that can affect the behavior or movement?
Yeah. I mean, this is obviously a very general question about it, that you could ask for any multi-stage learning system. In the ELL, I just showed you with that connectome that it's like all hardwired in there. So, in this ELL, the negative image it's learning is, it can learn complicated negative images, but this mapping, this across stages is pretty simple. The E cell goes to the I cell goes to the E cell. You just solve that wiring problem. It's a structural credit assignment problem that's just solved in the wiring. But then you're asking, well, what about more complicated things? What about, is that really how the cerebellum is doing it? Is it really just hardwired like that? I don't know. I mean, on the one hand, if you look at the cerebellum, it does have exquisite, incredible wiring. All of the literature on microzones and all of this incredible structure of the cerebellum, where you have these tight closed loops between the olive, particular sets of Purkinje cells are converging on the nucleus. Maybe that's the cerebellar version of this, and it is all solved through the wiring. That would be the easiest to understand, but maybe not. Maybe, but then you also might, the counter argument to that would be, okay, yes, that's true, but then, the cerebellum can't be doing anything. That would seem to restrict, the flexibility of or the power, I would think, of the cerebellum, as a learning machine, if it was all just hardwired like that. But I don't know. We don't know, I don't think. But that's, that's a good question. And even in the ELL, we always worried about that, just in the sense of, how do you even, in the ELL, how do you know that the MG cells are learning exactly the right thing to send to the output cell? Because it's not the exact same. They're not, why wouldn't you, and this is the question that bothers me a lot, is this why, when I wrote in the slide, why do you do it in 2 stages in the ELL? It seems like it would be better just to do it in 1 stage, because then you'd be guaranteed to have the perfect negative image. That to me is the most puzzling and kind of upsetting thing about all of this, is you see the circuit has this, clearly the MG cells are doing it. They're 90-something% of all the spines. They're so prominent. They're doing the right thing, but why does the system want to do it that way? We don't have a really compelling answer yet to why do you divide this into 2 stages and have all this elaborate circuitry, when it seems like you could do it all in the output cell in a better way. Clearly we're not, we must be missing something. But we don't know what that is yet. And if we learn that, maybe that would be relevant to the cerebellum too.
Yeah, that's a very interesting talk. Thank you so much.
Yeah, if you figure it out, please let me know.
Sure. Thank you.
Sorry, I know we're running late on time. Do we have time for one more question?
Fine with me.
Yeah. All right. I just have a quick question. So on that same line of thinking, in the cerebellum proper, we have Zebrin, which kind of indicates different P cell properties, and also kind of shows us some of the way that the olive projects, different parts of the olive project back to the Purkinje cell layer. Is there something similar in the ELL that shows how these sensory, or these, like what we're calling teaching signals, are topologically organized within the ELL? And does that also map to different ELL P-like cell properties?
Yeah, we don't know. Zebrin is in the Zebrin, actually, where they got Zebrin is actually from grinding up electric fish brains, interestingly. So that's where they cloned that, because there's so much cerebellum in these electric fish brains, I guess. It was easy to get. But in the ELL, that's in the cerebellum proper of these fish. In the ELL, it's all there's no zebrin. It's all zebrin negative in the ELL itself. And we don't have, we don't have a great amount of molecular profiling. So I don't know in the ELL if any of these Purkinje-like cells are molecularly distinct, or if there's molecular subclasses. I showed you today that there's these anatomical subclasses that you could kind of think of. You could think of these 2 channels I talked about as like zones of the cerebellum. But unlike the cerebellum, where they're kind of beautifully laid out in these sagittal stripes, in the ELL, they're just all kind of mixed up. And only with the connectome can you see how everything is actually ordered, so that there's a ton of order, as you saw in those connectivity matrices. It's highly ordered, but it's not laid out nicely like in the cerebellum with stripes or bands or anything. So I think that's all I can say about that.
Great, thank you so much. All right, I think we're done with the questions as well. Thank you so much, Dr. Sawtell, for the amazing talk, and thank you so much, everyone, for the questions. I hope you guys have a wonderful Tuesday, and goodbye.
Great, thanks a lot. Bye.